All right, welcome to the recording for RNAi and uh, control of genes, right? It's kind of a grab bag of different stuff, but this will definitely help you out for your little mini test that you're going to take over this. So one of the first things that you definitely have to know, because this definitely shows up on the AP test, more so in the last couple of years compared to maybe 10 years ago, is how genes can regulate based on operons, right? So what we have here is a operon. This is specifically the lac operon. Number one, this is your RNA polymerase. And RNA polymerase, remember the goal of RNA polymerase is to make a transcript, specifically mRNA, and it needs to attach to the promoter region. So number three is the promoter region. That's what you see it trying to attach to. Number four is the new piece that we're kind of talking about, which is the operator. And the operator is the governor, for the most part, of the genes that we're trying to transcribe and translate. So I'm trying to talk and write at the same time. Doesn't work very well. So this piece right here, number two, is the repressor. The repressor attaches to the operator. Spell this wrong. So the repressor attaches to the operator. You can see that right now RNA polymerase cannot move down this transcript, even though maybe 6, 7, and 8 are DNA segments that code for activating the enzyme lactase. Remember, ACE means enzyme, so lactase must work on lactose. Lactose, when the glycosidic linkage is broken, turns into glucose, which is readily metabolized, and galactose, which we can use isomerase and change it into glucose. So the goal is we have to make lactase, the enzyme, to break down lactose. Well, we're not going to break down lactose unless the environment has lactose in it. So operons are really good at helping organisms regulate needs when the environment presents different types of situations. So if lactose is in the environment, the bacteria, in this case, might want to break down lactose. It needs the enzyme lactase to do that. So here we have an inactivated repressor by lactose. So lactose in this scenario is the inducer, right? It acts in inducing the transcription and translation of this operon. So it acts as the inducer. Now, there's more to this that we have found out over the time, that there's actually two pieces that kind of govern how this works, because if lactose is present and glucose is present, this will govern how much lactase actually gets made. So there's other pieces, specifically CAMP, cyclical AMP. AMP stands for adenosine monophosphate, which can be, this is a reversible reaction here, turned into ATP. And ATP, when it gets used, turns back into AMP. Since it's called cyclical, that must mean that in the life cycle, it cycles in the amounts inside the cell. So cyclical AMP attaches to catabolite marker piece that is called CAP. And when CAP and CAMP join together, they attach right here at the promoter region, allowing RNA polymerase to come in. So the idea is if glucose levels are really high, well, that means the organism is probably making lots of ATP, right? So the ATP concentration increases, which means that AMP will not be high, so CAP will not attach, and RNA polymerase won't come in. So there needs to be two things in order for us to make lactase. One is to have glucose in low supply, right? Reverse this idea. It's got to be in low supply. If it's in low supply, then that means that cyclical AMP is in high supply. Since cyclical AMP is in high supply, combined to CAP, allow RNA polymerase to attach. The other thing that needs to be present in order to make lactose or lactase is that lactose needs to be present, just like we stated at the beginning of this. So these are two things that allow for this operon to actually govern how the cell is going to respond to the environment, in this case, sugars that are present. Another example would be activating a repressor based on the environment. So we talked about tryptophan, so let's write trip. And trip, when the concentration of trip is high, trip activates a repressor, which then binds to the operator. So if there is not a lot of trip in the environment, the repressor will not be on it, and we can transcribe and start making the protein tryptophan. But as the concentration increases, the repressor gets attached to and put onto the operator, and then we block RNA polymerase, so we stop making tryptophan. It makes sense. Once your concentration gets up high enough, we want to shut it down, right? If the concentration is low, then it should be open. So it's a way to kind of like regulate how much of a specific product ends up getting made. So this is operons. All right, next piece. This is just showing that cap and the CAMP that we were just talking about. Cyclical AMP binding into cap, activating and allowing RNA polymerase to bind in. But once again, it has to be if lactose has pulled off the repressor, because lactose acts as that inducer piece. All right, let's talk about epigenetics. So that's a control based on the environment. Another way that organisms can control transcription, translation, and turning into a phenotype, because remember we go from genotype, which is the DNA, to phenotype, which is your actual proteins. The whole goal is to get from here to here. So we talked about how the environment can allow for this genotype to turn into a phenotype. Here we're going to talk about epigenetics, which is the control of acetylation and methylation. So in this example, we have histones that are all bound together in this nucleosome. And right now we can't get at this DNA because it's too tightly packed up. Well, based on environment, what you consume, acetylation can happen, and it'll cause spacing between these histones. And now we can actually get in at this DNA to transcribe and translate it. So this is acting on the genotype directly, right on the DNA. The other one is called methylation. Watch that fantastic NOVA video about methylation and those rats, and how the Goody gene, if it was fed a high diet of methyl groups, the Goody gene was not activated, and in turn didn't allow for the mice to get fat. So methylation can be passed down from mother to sons and daughters, even from grandparents on down the line. But these all come in from the food you eat. And what happens is it takes pieces of DNA, so this is a piece of DNA, the methylation will deactivate this part of the DNA for the time being, not allowing it to be transcribed or translated. So epigenetics, epi, surface, genetics of your genome, is all about controlling your genome at the surface, right, before we actually do transcription translation. This is one of the reasons why identical siblings end up looking different throughout life, is because they consume different things, methyl groups and acetyl groups, that change how their DNA is transcribed and translated. All right, next thing we need to understand about Hox genes, or controller genes. They're mostly for body plans. They are highly conserved, meaning that from one organism to another, we tend to see similar genes. Now, whether or not we get duplications of those genes or slight changes in the gene sequence is a different idea. So you have your genome that codes for the proteins, right? And we talked about how there's a very small portion of your genome that actually codes for proteins. We used to think that the rest of it was junk DNA. That's not true at all. A lot of the rest of your DNA is coded for all the other different types of RNAs that are important, and they could be these Hox genes that are controller genes. So these things allow for transcription factors to attach to allow us to do transcription, which are important for producing the protein. So these actually govern whether or not we can turn the phenotype into something later on. So if any of these Hox genes get changed or duplicated, there'll be dramatic effects because these are so important. They're controllers. An analogy I can give you, which is similar to the epistatic thing we talked about, would be if you plugged in a light bulb. If you plugged in a blue light bulb, and flip the switch, it would show up blue. But what if you didn't flip the switch? Even though you plugged in the blue light bulb, you would never see the phenotype of the blue light. So the light switch acts like the controller gene. It controls whether or not you actually see the light. All right, next up, RNAi. I'm going to give you the simplest version so that you understand what's going on here. It stands for interference. So this is not stopping it at the genotype, but we're going to stop it when we go from DNA to RNA to protein. In epigenetics, we control it here. In RNAi, we control it here. We can stop the RNA from turning into a protein. And the way they do it is double-stranded RNA gets cut by a complex called dicer, and then we make small interfering RNAs because of it. So one side is actually going to bind into the transcript that we're trying to silence or stop. So dicer will cut it, hence it's dicing, it's chopping, chops the double-stranded RNAs into two pieces. One of the pieces will be considered the silencing RNA, and then RISC is the other piece that's going to help us attach it to the mRNA or help us find the mRNA that the transcript that we're trying to destroy right here, and then we'll chop up the mRNA. So if the mRNA all gets chopped up, it never gets to the ribosome, we never turn it into a polypeptide chain, hence silencing the whole process. So when we watched the video, there was a cop that was chopping up these double-stranded pieces or these transcripts. The cop is actually a complex of these two segments, RISC and dicer, together. So those two pieces really need to work together in order to silence our transcripts to turn into protein, and that's RNAi. Right, so we've got these microRNAs or miRNAs, or silencing interference RNAs, double-stranded RNAs. The big thing is that you understand that, here we have this double-stranded RNA, dicer chops it, we end up getting small interfering RNAs now, and those small interfering RNAs can be used by RISC to find the transcript that we want to destroy. So for example, if this is the protein transcript for Huntington, which is the bad protein that gets made by Huntington's disease, we can make these double-stranded RNAs to look like that protein, inject it in, dicer will cut it up, make one segment of silencing RNA piece, RISC will find the bad transcript for Huntington that builds up, Huntington that builds up in a person that has Huntington's disease, and instead of, even though they have a defect in their genes, we can destroy their buildup of this protein, Huntington. That's a way of silencing. So this is one of the new medical breakouts, and we're looking at this. We watched in the video how it helps with macular degeneration, cancer treatments. There's a lot of different things that this has possible applications for. All right, stem cells. We need to discuss these real quick. What it means to be a stem cell. So stem cells are cells that have the ability to flex or change. They have plasticity. Now there's two different types of plasticity. There's multipotent and then there's pluripotent. Multipotent means that it can change into a variety of cells. So an example would be bone marrow can turn into all the different types of blood cells. Right? So you have red blood cells, you have white blood cells, you have platelets, right? All found in the bone marrow. We consider those adult stem cells, and they're multipotent. You can't take a bone marrow cell and turn it into a neuron. That's just not how it works. So those stem cells are somewhat determined, but not fully determined at that stage. A stem cell that has pluripotency is like what it's showing here, where we have a cell that can turn into any kind of cell. And the cool new advancements, and this is a Nobel Prize by Yamanaka, is that we can do what's called IPS, or induced pluripotent stem cells. In the past, we were able to do a cloning device where if you took a cell and we did a somatic cell nuclear transfer, so we took out the nucleus from maybe your diploid cell, and we took an egg, it has haploid amounts, and we remove the actual genetic material, take that diploid content and put it into this egg, re-put it back into mom. Mom will then be able to grow this clone of you inside her. Right? Kind of creepy. But this was identical twin type of deal is kind of the same way that that's working. We have two cells growing in tandem at the same time if they have the same exact genetic information. We would look at this not to make clones of ourselves, which we have made clones of animals, but more for therapeutic cloning. So cloning for maybe the cells. So as the cells start developing, embryo-wise, there's a blastocyst that forms as it develops. So it starts as maybe 2 cells, and then the 2 cells form into 4 cells, and they get up to 8 cells. And eventually they form this hollow ball of cells, and at this top layer up here, these are considered the embryonic stem cells. Embryonic stem cells are pluripotent. They could turn into any of these kinds. Right? Obviously, there's a moral dilemma about this, because once you take these cells away, this embryo no longer develops. If we left them in there, they would develop into an individual, a human. Right? So there's morality things there. Are you killing something that's alive? I don't care your opinions necessarily on this. I'm just giving you the fact pieces. But iPS cells, they have this pluripotent piece as well. But what they do is they take the adult stem cell from you, and using reprogramming factors, pretty much a retrovirus is injected, but they have new treatments now. Yamanaka did this. Use a retrovirus and ended up developing 4 programming or reprogramming gene activation, which makes the cell act young, and those are called Yamanaka factors. Those Yamanaka factors make the adult stem cell, or the adult cells, back into an embryonic-like cell, a cell that has plasticity, so it can flex into any kind of cell. And the cool part is, you could then use these cells, since they're your DNA, for any sort of transplant. So you have tissue issues in your lung, you have blood cell issues, muscle issues, nervous cell issues. We can do all those types of things by placing then those iPS cells, once we give them some chemical factors to differentiate. You can add some chemicals to make them differentiate into any kind you want. We can then put them back into you to fix you with your cells. So there's very little organ rejection, because the DNA is exactly the same. It is your cells, just reprogrammed. So that's stem cells, and the extent of what stem cells has the possibility of fixing. The last thing that you need to understand is cell signaling and how cells talk to each other. We've talked about this a little bit over the course of the year, but you need to know the 4 different types. So endocrinal, this is when a cell releases stuff like hormones into the bloodstream, and then a receptor cell. So we got these glycolipids and glycoproteins, these things that are embedded in the cell membrane that allow for the reception. Neuronal, where we have like a synapse. So this would be synaptic, where we secrete a neurotransmitter, affects the second cell. Paracrine, where we have one cell releasing, and we end up getting local deployment, right, to the cells that are very close to it, causing the target cells to change in some way, shape, or form, or react. And then contact, when one cell has to touch another cell. Regardless, they all have the same idea, in that we have this reception, transduction, and then an intercellular response. So here's your cell signal. Maybe it's insulin, attaches to some sort of integral protein, maybe, allows for maybe a port to open up, maybe something comes inside, or it causes a transduction pathway on the inside. And this transduction pathway, like a cascade, a waterfall, allows for an internal cellular response. Maybe it's transcribing and translating, maybe it's DNA replication, maybe it's making more organelles. Regardless, we have this whole thing happening. The way to stop it then, right, we have this, it'll cascade and keep going, keep going. The way to stop this then is to destroy the signal molecule, right? You don't want to destroy the receptor, because then you have to repair and make a new receptor. But if we destroy the signal molecule, now the whole transduction and response process stops. So an example with a neuron talking to a target cell, maybe this is a neurotransmitter like acetylcholine, right? Acetylcholine causes skeletal muscles to contract. So acetylcholine is secreted into the synaptic cleft here, and then it's received at the end by some ligand receptors, and it opens things up to allow molecules to move into this cell. If we don't destroy that acetylcholine, it'll keep the action going. So there's enzymes that could break down acetylcholine called acetylcholinesterase. That could break it down. things can diffuse away and allow for it to stop. There's a lot of different ways that we can stop this actual cell signaling process, but you definitely need to understand how one cell can, air quotes, talk to another cell, right? Make another cell respond in some way, shape, or form. So keep this idea in mind. There's a question or two about this as well. Otherwise, I think that summarizes and helps you out enough for your mini test, and we'll come back and watch this video probably when we get closer to AP test time. Good luck.